Hello, my name is Jeremy Elmore, and this is my presentation for LUT1 with Western Governors University. Today I'll be presenting information on genetics. Let's start off with asking a question. Were you aware that a discovery made approximately 60 years ago has the ability to possibly eradicate disease and physical ailments of humans today and in the near future? Research suggests that since the discovery of the DNA double helix model by Watson and Crick, the advancements in genetic research and genetic engineering could make disease and virus eradication a viable possibility due to mapping of the human genome and the discovery of the vital role siRNA plays in silencing of specific genes. The study of genetics is a fairly recent scientific concept when you look at the history of science. For the most part, the study of genetics began in the late part of the 19th century, whereas the study of astronomy, physics in some form, chemistry, or alchemy have been experimented with for centuries. Advances in technology, such as higher-powered microscopes, more sterile and secure ways of handling animal blood and tissue samples, led scientists to look at what was in the blood for deeper meaning. The identification of varying blood cells, the white and red blood cells, and their purpose or function in the species, then led to a deeper dive into what was in the cell itself. Friedrich Miescher in the 1860s was the first to identify inside white blood cells what would later become known as nucleic acid. Scientists found that inside each cell was a complex working of molecules that had never been seen before. Cells had a nucleus which housed an abundance of molecules that each performed their own unique function to make life possible. The discovery of DNA, or deoxyribonucleic acid, by Miescher was a huge breakthrough in the science of genetics. Researchers had now moved from studying the blood itself to what made blood the carrier for species identification, and the variances from person to person or animal to animal was due to the unique genetic makeup of the DNA. Perhaps the most widely known and publicized finding that has led to so many advances with genetics is the DNA double helix model of Drs. James Watson and Francis Crick, for which they were later awarded the Nobel Prize. This discovery led them to create the model of the DNA molecules we know it today, basically a spiraling ladder of genetic material of 4 bases: A, adenine, C, cytosine, G, guanine, and T, thymine. These 4 bases, when coded together in a chain, form the basis for all genetic code in living organisms. These bases also work in tandem. A equals T and C equals G. Perhaps one of the most significant research projects since the DNA double helix model was introduced is the Human Genome Project, which was completed in 2001. The U.S. government and other national governments, along with private research institutes and public universities, set out to map the entire human genome. To simplify what a gene is, it is basically the result of the A to T and C to G pairing of DNA stretched out over hundreds or thousands or millions of bases in a linear string. Researchers first had to design millions of gene fragments and have these specially synthesized, where each gene fragment, typically 70 to 90 bases in length, were created in silico, or by computer analysis, then arranged them so that they stretched to form the estimated 6 billion base length. As the life science community was just beginning to synthesize DNA sequences at this length with the required purity and specifications to meet the stringent demands of this project, it was quite a feat that the project actually concluded prior to the estimated date. At the conclusion of this project, it was discovered that the human genome was 3 billion bases long, half of what researchers had originally estimated, with some 32,000 unique genes. Since then, researchers have been trying to decode the human genome to see what information it does or does not contain. It was discovered that approximately 80% of the human genome is considered junk DNA, or does not have a direct impact on genetic makeup. The significance of this project and what it uncovered should not be underestimated. Findings about what function DNA plays in the role of genetics, while previously known to be significant, was now revealed to be more essential in the function of gene regulation than simply the code. DNA houses the blueprint for all living organisms. All humans follow this blueprint, while slight variations in the DNA code produce the physical variations that make each human unique. It is this template-dependent law that keeps us similar while allowing for variations. Another significant breakthrough was the discovery of the RNA molecule, ribonucleic acid, in the cell and its interaction and significance in genetic coding. Since the discovery of RNAi, the implications of RNA, siRNA, shRNA, mRNA, and miRNA, all of these more commonly referred to as RNAi, and their interactions with protein enzymes have led science to see that these interactions are so tightly bound to each other that the possibility of one molecule telling another what to do or not to do has shown that science can manipulate the genetic code. The phenomenon of gene silencing is now referred to as RNAi. Instead of mopping up water from the floor, RNAi works by turning off the spigot. Just as diseases or viruses must have a code, then they too need to be studied to find out how best to stop their replication. If cancer is used as an example, which is one of the most prominent diseases in the world, it is the result of a single cell mutation, which rapidly divides and causes other mutated cells, thus leading to tumors. If researchers can find the reason for this mutation, could the mutation and replication codes be turned off? This is what researchers are looking to do with disease of all types. Research at the cellular level has shown that each cell has identical genetic material, yet the way this interaction takes place in tissue and at the protein level is where researchers and pharmaceutical companies are now beginning to understand how best to use siRNA in conjunction with the protein. The unique role that mRNA or messenger RNA plays in the genetic process is simply that of a messenger. It carries the coded message from the DNA to the protein, and the protein then uses the code to perform the associated function. Research is showing promise in using RNAi as a therapeutic in some cases, but the end result truly needs to be that the mRNA carries the code to the protein molecule to truly eradicate an element. It is this code that research seeks to decode for each disease in each patient. In conclusion, over the last roughly 60 years, medicine and research have seen a tremendous shift in what may be possible for humans and their overall quality of life. Watson and Crick's breakthrough discovery led us to the Human Genome Project at the turn of the century, and in just over a decade, science is on the verge of everything from selecting the eye color of our children to preventing disease based on our genetic code, and what science can turn on or turn off based on recent siRNA discoveries. Our children and grandchildren may also have the options of being genetically coded to make sure diseases such as mental retardation, muscular dystrophy, or genetic predisposition for heart disease, among many, many others, can be turned off before birth. Questions?
Yes, my first question is, if Masur discovered nuclein, why were Watson and Crick credited with the discovery of DNA?
Many people make that mistake because of the overwhelming popularity of Watson and Crick's DNA double helix model and the lack of information or discussions about Masur. Masur did discover what we now know as DNA. It was Watson and Crick who discovered the DNA double helix model. Next question.
Question number 2. Could you describe the way DNA and RNA and proteins work together in a simple manner?
Sure. Everyone is familiar enough with the electricity of their home and how it functions. Massive amounts of electricity flow to your home from an electrical station, in this case DNA. In your home, the light switch is RNAi, and the lights are the protein enzymes. Regardless of what lights or appliances are in your home, the same electricity or DNA flows to it. Now, when you need to turn on a lamp, you have a switch, RNAi, that must be controlled or send a message to the lamp, the protein enzyme, for it to function properly or not function at all. Any other questions?
Yeah, my final question is siRNA currently being used in the prescription drugs that doctors prescribe to us?
In most cases, no. siRNA needs a transfection vector, or a vehicle, if you will, to insert itself into the cell. Oral medications are not good transfection vectors and thus limit the effectiveness of the siRNA therapeutic. Intravenously administered drugs are a more suitable method for siRNA therapies and have shown promise recently of being effective in some cases.